In Illumina, amplification is performed by, we call it as solid phase PCR, where these fragments are amplified on glass slides. The amplified same fragments which are amplified, they make up those what we call them as clusters. So we have a large number of clusters, around 40 million on those slides, and where we can have over 1,000 copies of each single fragment in each cluster. Why we amplify them? Obviously we need to detect a stronger signal. So in order to have that, we need to amplify them. So this is obviously the one step which is kind of additional step in these next generation sequencing machines. Let's recall the amplification in Illumina. We have seen that the fragments, they get attached to the base of the slide with what we call them as adapters, and then these adapters on the other side, they are complementary to the primers which are also located to some neighboring positions. So then those primers, they attach or they hybridize with these adapters, and then they amplify the fragments in an arrangement which we call it as a bridge amplification. So a lot of bridges, then later on returns different clusters. So we have these all clusters which are representing the amplified copies of those single molecules. Illumina uses reversible terminators which are labeled by different fluorophores, different molecules that give specific colors for all the 4 bases. So we have different colors for T, different colors for G, C and A. So here we see the terminators. Terminators are special nucleotides. Why we call them terminators? Because they terminate the growing chain of the DNA. Once they get incorporated into the replication process or this sequencing process, they cannot continue further. Reason? Because if we look into the structure of the sugar, we see on 3' position, or the position number 3, carbon number 3 of the sugar, there is an nitrogen group which is attached, and because of that they cannot accept the new nucleotide. So after this nucleotide is incorporated, the chain cannot grow further. Same way we have the fluorophore molecules which are attached. So for example, there is a green color attached over here. We can have red for other nucleotide, and we can have blue and yellow or anything else. So those indicate that which nucleotide has been incorporated. So here is the terminator after the fluorophore molecule is been removed, as well as the termination group is also removed. So we add this terminator, then fluorophore gives the signals to the camera, and then after that we remove the terminator group, and then it's ready to accept the next or new nucleotide. So in this way then the chain keeps on growing, and we keep on recording the sequence one by one. So in Illumina we process all 4 labeled terminator nucleotides which are differentially labeled with different fluorophores. We process them all together, and then different bases are located into different nucleotide fragments or reads, and once those bases are incorporated, there is the release of fluorescent signal which then is been picked by the camera. So there is an imaging, and after that the fluorophore and the terminator groups are removed, and the process keeps on repeating. This is how we can see that again. Here are different nucleotides which are coming. We see that these nucleotides, they get incorporated into these reads, and in the next step we see that fluorophore, after generating the signals, they are ready to being relieved. So fluorophores are gone, and then obviously the terminators are removed, and we see that these 3 reads then are ready to accept flow of next 4 bases. Again the fluorophores are removed and cycle continues. So let's see how camera sees them. So here in these diagrams we are focusing on 2 clusters. Number 1 is this guy at the top, and number 2 is this one. So we see that these 4 nucleotides are differentially labeled. C is green, A is blue, T is red, and G is yellow. So if we concentrate on the topmost cluster, so at first point there has been an incorporation of a C followed by A, then we see a T here, red color, again a C and a G and a T. So that is how this sequence has been read. And in the bottom cluster we see there is a continuous incorporation of C's. So this is how camera sees them and keeps track of indexes or the positions of those different clusters and keeps on recording each position and change in color in each position. So in this way we can get the whole sequences recorded in the shape of these CAT, CGT or CCCCCCs. These are actually the reads which are in the end produced. So in the end we conclude that Illumina performs sequencing by synthesis reactions using terminator nucleotides which are differentially labeled with different fluorophores.